Oh, it's down there. Oh, hello, recording.
Got it.
And so we just kind of think about it as microbes being everywhere underground. But that's not actually true, right? It's not that all microbes are everywhere. We do also have a perception of certain fungi belonging to certain places. And I think, right, this is one of the most clearest ways we see that is a lot of funga are grouped by what area you might find them in. And that can be, by human definitions of country, continent. But we actually do have that concept. And maybe it's just time to apply this concept to invasive thinking. Anne told me this at the beginning of my PhD. She said this was kind of the theme of her lab and a huge message that she is trying to get out in the last maybe like in this stage of her career, is that every place has an endemic microbial diversity, which is at risk of being displaced or thrown out of balance from its natural functioning when non-native species are introduced. And so I think that this is kind of a legacy that Jason, me, and all of her students right now are trying to carry out. And yeah, it's also kind of a simple thought, but it's one that really has stuck with me now, and I try and spread it as far as I can as well. All right, so let's get down to golden oyster mushrooms. These are wood decay fungi. So there are many different classes of fungi. I think you all are probably some amount familiar with that. There can be fungi that decay um, live plant tissue, or maybe leaves and other dead matter, or ones that partner with live trees like mycorrhizal species. But golden oysters, they decay dead wood. I've heard I've read and tracked down a claim that they are slightly pathogenic, but I have never found any publication or research to back that up. I traced it back to like an early 1900s like Nordic text about fungi, and that was the first time it was ever mentioned, but it was not in relation to any research. So I don't know if it's pathogenic yet, but I've mostly only seen them on dead trees. Wood decay fungi are the primary decomposers of the carbon cycle, right? So, like that step where carbon has been locked up into wood for hundreds, maybe thousands, millions of years, fungi are the most, and white rot wood decay fungi specifically, which golden oyster is a white rot, they are the most efficient organisms at unlocking the carbon that's been trapped in wood and releasing it back into the cycle. And they can release it either into soil, they either capture it and integrate it into their own bodies, or they exhale it as carbon dioxide. And so understanding how an invasive wood decay fungus might affect this decomposition cycle is really important. If it is that pivotal step, what happens when that step is thrown out of balance by an invasive fungus? We don't know yet. So there are also examples of invasive mycorrhizal fungi. On the left is Areoboletus projectilis. It's right, kind of like in the class of boletes. And then we also have the famous Mario mushroom, Amanita muscaria, and it's on many continents and associated with pine plantations. So there's definitely human mediated movement involved with some of these other invasive fungi as well. There are other examples of invasive wood decay fungi. This one you all might have heard of as dry rot. So this is Serpula lacrymans, and it can cause really heavy structural damage and has caused millions, possibly even billions in economic damage in multiple currencies across the world. There's also Flavidon subulatus, which has been renamed actually to Urpex subulatus. That was introduced by an invasive partner beetle. So it was introduced by the ambrosia beetle to the southeast of the United States. And this paper, Husino et al. 2020, showed that in like a controlled log experiment, when they inoculated logs with Urpex subulatus, it displaced native fungi. So they were able to document that ecological damage in like kind of an experimental setting. So now the golden oyster is from a pretty broad range over in East Asia, pretty much all the way from the southeastern border of Russia, all the way down to like Taiwan and even like parts of the Himalayan mountain range. And so you can kind of see that it's more or less along this 40 degrees north latitude, where it has landed up also in North America. And how did it get here? We pretty much think a big part of it has to do with the fact that golden oysters, which I might call GOM from now on, are edible, right? We can cultivate them, and it immediately is a product, right? It sells itself. And so I have read in Paul Stamets's 2000 book called Growing Medicinal Mushrooms. He documents that he brought it over from Asia in three slants. He kind of even uses the language like he snuck them over, which I think, I don't know, just an interesting choice of self-incrimination, I guess. But we don't know whether it was really him or not. But either way, golden oysters came over because they're cultivatable and they're delicious. And now they're sold in these mushroom grow kits, right? Like pretty much all over the world, but it's especially become a popular product in the US. And what's kind of funny is like we look at this mushroom kit and we don't immediately think of it as a biotechnology, but that's kind of what it is. And it's important with emerging biotechnologies to kind of consider what are the risks that it poses. But kind of as per usual, as is the course with human history, is we don't assess risks first. We sell the product first. But we learn our lessons. So we'll see about, we'll see how it goes from here. These are pictures of gum that I've taken in Wisconsin. This tree on the left, I nicknamed the Beacon because I saw it, gosh, I can't remember which park around here where I saw it, but I saw it from like, I want to say just over 100 meters away. I mean, I was really quite far away and I couldn't believe that I spotted it. It was just like shining yellow, you can spot them from far away and it is arresting to see them in the forest. I have no doubt that each of you has experienced the same thing where you're just like, oh my gosh, look at all these clusters of golden oyster. When you come across them, they almost kind of give off that feeling of being weedy and invasive. And I think that's a really curious like feeling and intuition that I like to think about and I like to hear about other people's experience with it. Let me play this video here where you can see
Yeah.
millions of spores being released. I mean, it's just crazy, right? That's just 8 seconds of it releasing spores, and that it was probably in the millions. And that's just 8 seconds. As you all know, they fruit for months repeatedly in multiple clusters, and they usually stay there for a few days. Um, so that is kind of crazy to think about too. So the questions we're going to talk about here today is how quickly has GOM spread in its invasive range of North America? And the money question, is GOM displacing native fungal communities? So let's get into this first one. How quickly has it spread? So I started with the year 2016 with these first 5 observations in Iowa, Illinois, Wisconsin, Michigan, and New York. I've heard from some folks in Iowa that the first time that they actually saw it on a foray, I think it's like the Iowa Mycological Club or Society, they saw it was in 2010. But I only had online databases like iNaturalist and Mushroom Observer to go off of, so that's where I pulled these orange data points from. So the orange data points are going to be per year, but the yellow filled out states are going to keep track of cumulatively which states it has reached so far. So we start in 2016. 2017, it's definitely getting a foothold in the Midwest. It's spread into Ontario, first Canadian territory. Makes a jump over to Colorado and really starts to fill in the states in between. And then we arrive at 2023, which was the last year of the data set that I included. And yeah, it's really just insane to see how quickly it spreads and again, this is only per year. When you go on iNaturalist, you get an even more kind of like you get a wall that hits you because you see all the cumulative observations of where it's been over time. So it's really spread quite a bit across the Midwest and Northeast, and it doesn't seem to be stopping. I've noticed but from 2024 and 2025 that it has also claimed North Carolina and Alabama. And I have I met someone who was from Kentucky, and they said that they found it there as well, but no observations have been posted from Kentucky yet. So, how quickly has it spread? I think it's fair to say really fast, in just 8 years. So, let's go to the next question. Is GOM displacing native fungal communities? So the way we went about answering that question was we took wood shavings from inside trees that had a golden oyster cluster on them and trees that did not have a golden oyster cluster on them. Now you must be thinking, well, not seeing a cluster on them isn't foolproof, that there isn't still mycelium inside, and you're exactly right. But it turned out that there were only two trees that we misidentified in that way, which kind of it really begs the question like, wow, so like how quickly does golden oyster fruit on a tree to where we only got two of them wrong, Like seemingly it takes any opportunity once it's gotten into a tree to fruit and reproduce. So it was great fun going out into the forest, putting ladders up against trees and getting all of these drilled shavings. And then once we get those drilled shavings, we extract the DNA from them with some cool chemicals. And then we use a kind of more new type of sequencing called high throughput amplicon sequencing. So we target a section of DNA that is only in fungi. So we're not going to get any insects or like the plant DNA that might have been in those wood shavings, and we get the DNA of the entire fungal community, like any of the DNA that was amplified from the fungal community in those tree sections. And then we do some bioinformatics and we figure out, okay, who was where, which fungi were in which samples. And so I'm so sorry to not share my actual data, but I'm like a month out from publication, so I'm just trying to be a bit protective over that, but I will surely send all of you my publication as it comes out. But basically my data looks a lot like this. It looks like the Husino et al. Paper where they found that Urpex subulatus does cause that ecological change. We found the same thing with golden oyster. So on the left, you see a box plot where the control, in our case, trees that didn't have GOM, their fungal species richness, which is like how many different fungal species are in that sample, it was significantly higher than the trees that did have GOM. Their species richness or the fungal diversity was much lower. And then this plot on the right with the circles, it shows community composition. So it's kind of like if fungi A, B, and C are in one community, but fungi X, Y, Z are like, it's a completely different composition. Who is there changes between trees with GOM and trees without GOM. So that that community composition is significantly different when GOM gets into a tree. And the kind of crazy thing too is that, in Husino's inoculation experiment, they really plugged that log full of the fungus. We found these same results in a natural setting, without shoving the trees full of GOM. That is to say that GOM got enough of a foothold into the tree on its own, colonized so much of it so as to see similar results as a inoculated experiment where they really slammed the logs with a different invasive wood decay fungus. Um, Okay, so this is kind of a finer point of the experiment that I thought was kind of crazy. We got a snapshot of 7 trees in which not all of the heights were colonized. So like we sampled 26 trees total, but 7 of those trees were in intermediate stages. That is to say, it was not, we didn't detect GOM at all 3 heights, or we didn't not detect GOM at all 3 heights. So there, right? We might have found like GOM in 1 or 2 of the heights, but not all 3. And the thing that was particularly crazy here is let's just focus in on D. We see that a height in a tree that has GOM is more similar in its fungal community composition to entirely different trees that have GOM in them compared to the other heights in that same tree that did not have GOM. What I kind of mean to say by that is imagine a single tree and you have a bunch of fungi that have gotten into that dead tree, right? That single tree unit, it's kind of intuitive that it's going to have the most same fungal community composition through like within itself than compared to a entirely different tree, right? GOM flips the script on that. So it changes the community so much that that height then becomes more like entirely different trees that have also been colonized by GOM. It's more similar to them than it even is similar to other heights in its own tree that have not been colonized by GOM. And so that was a really crazy finding. And then the other kind of intuitive conclusion to that is that GOM's effects are spatially related, but we expect that of any fungus, right? So it's just kind of reinforcing that when it gets into a tree, it's not that the entire tree is a goner right away. It does, in fact, have to colonize that area, and then it has spatial effects on the fungal community that is sharing the space that it has colonized. Um, yeah, so just to reiterate, GOM seemingly enters a tree in one spot, affects the community in that immediate area, and then it grows out to affect other areas and heights. This is actually the example from the previous slide, but it was kind of an interesting part of our sampling is we had one tree where GOM had only gotten in on the roots, and you'll notice I've circled in red the spots that we sampled, and it just happened to be on either side of the column where GOM was growing. And we actually did not find GOM in these wood samples that I've circled in red. So it was kind of an interesting look at the really early stage of GOM getting into a tree. Wood decay fungi, they grow along the natural pipes that the tree has laid in, right? So all trees have vasculature, and that vasculature runs up and down, right, in the direction that the tree grows. And so golden oyster and most other wood decay fungi, they use that piping first to go up, and we just happened to miss it by drilling on the other sides of it. I've had some really cool experiences at the UW Arboretum, and I'm really grateful that they have funded me. Where people come up to me. So if you see this gentleman in the gray and yellow, he showed me Hypsizygis ulmarius. It's sometimes called the elm oyster. He showed me a specimen from it of it that he has from years ago and said, like, I never see this mushroom anymore. Like, can you find out what's happened to it? And I was like, that's a really great idea. And that conversation single-handedly like made me do an extra analysis on my data. So I went to go look at what fungi do we find most often in GOM-affected and unaffected trees. And so you can see on the left here, you obviously find GOM, some trichoderma, the dryad saddle, Serioporus squamosus, armillaria, the honey mushroom, also known to be a pathogen, and then Serena unicolor coming in at the bottom in GOM affected trees. But in GOM unaffected trees, Serena unicolor is the top. It showed up at the highest frequency in those ones, and then you see just a different roster of fungi in GOM unaffected trees. So that was really interesting, and I yeah went into my data and saw that Serena unicolor is the number one affected fungus. That is, its its prevalence completely plummets when you look at trees that have been colonized by GOM. So I'm kind of using it as my what's the word for it? My ambassador with with sad faces on it to show everyone like this is what we stand to lose, right? Is the wood decay fungi that we've always seen walking through the forest. They are under threat by the golden oyster. Where could GOM potentially establish invasive populations if it's introduced? So I used some publicly available climate data and this species distribution model program called Maxent. So Maxent, you feed into it observations where the species is currently found, and you feed in the climate variables, and it makes that association. Okay, for the climate variables in the grid squares where it's found, we use that association, then it applies it to the rest of the map that you give it. And so we found that Europe and kind of this entire orange central streak going through Asia would be prime climatic suitable habitat for GOM if it's established there, and then parts of South America too. And I will say also huge swaths of North America where it hasn't gotten to yet. Um, Very sadly, I'm kind of looking at the Pacific Northwest and it seems like little bits of Mexico too, and I think I've heard that there is great fungal diversity in both those places. And the wild thing at the end of all of this is it's still, foraged and sold in Wisconsin. You can buy it online and ship it anywhere in Australia. It's been naturalized in Italy, and it is used as an agro waste processing material in three other continents, right? South America, Africa, and Asia. So because golden oyster grows so fast and breaks things down, it's actually being used in a really creative way. Like all these other countries that have agricultural waste that needs to be processed and broken down, they're kind of using like a circular, sustainable way to process it. But unfortunately, it's with this fungus that has been shown to have some pretty great invasive potential. And so GOM continues to be sold and grown globally, and along with other non-native fungi as well. So the story doesn't even really end with golden oyster. If you look at this table, it really makes you wonder, where did these other fungi come from, right? Like, even if it's native to the broad area, what about that specific strain? Did it come from a different country, or has it been bred to be particularly aggressive more so than the native variety? These are kind of all questions that we're tossing around right now. Some future directions where I'm taking it. Unfortunately, I just kind of have taken stock of my PhD and how much time I have left, and I don't know if I'll be able to do how fast does GOM decay wood compared to native decay fungi. So, unfortunately, I think I'm dropping off that chapter. But then this middle and right chapter, Our populations of GOM diverging evolutionarily is underway right now. And then how do different groups feel about management? I'm about to start that chapter as well. So these right two chapters are kind of happening right now. You might have seen in other groups or listservs that you're on that I'm actively recruiting folks across North America to send me samples of golden oyster mushroom from all over North America. So I posted that in the North American Mycological Association listserv, and then a bunch of really kind folks from there have gone and posted it on other regional groups. And so if you know that you're traveling to somewhere like Kentucky or Tennessee or Ontario, Canada, please do let me know and keep your eye out for GOM. I have a project page that Jason very kindly helped me build that details all of the instructions that you can follow if you want to send some GOM. And then how do different groups feel about management? I'll pretty much be conducting like interviews with stakeholders from various groups because that is the proper way to start invasive species management, right? So my opinion on what I think should happen with the golden oyster situation, that's just one voice. We also need the voices of amateur mycologists, of foragers, of mushroom sellers, of people in government, right, who are in charge of deciding what foreign substances or foreign live material can enter the country or exit the country. We need a basically joint open conversation with all of these stakeholder groups, and together we need to all come to a um, compromise and solution together of how we should go about managing this invasive species. This is what helping can look like in mycology. I have no doubt that a number of you have contributed to the iNaturalist or Mushroom Observer posts that I used in my data. And I can't tell you enough how grateful I am. This project really would not exist without community science. I monitor it all the time. I have really dramatic moments where I'm like, oh my God, it's in Alabama now. And I click on it and I message the person on iNaturalist and I go crazy. So thank you for everyone that's contributed in that way. It means everything in our field. And then the other thing is you can also try some more responsible cultivation, right? So I've heard golden oysters can grow in your bathroom, so why not try that? At least it's not outside. So my parting words for you is spread the word, not the fungus. Golden oyster is now naturalized in South America, Europe, Africa, and India. India, I'm not quite so sure, is this a case of invasion? But the point is that the scientists and the community scientists in those continents will tell their story. And so, if you have family or you visit these areas, have a conversation. If you come across a mushroom market, somewhere in South America, like have that conversation. And hopefully, we can cultivate this idea that, we shouldn't move invasive species around and that fungi can be invasive. So, thank you all so much for listening. I really appreciate you all having me, and let's talk. I want to hear your thoughts.
Sweet. Thank you so much, Aishwarya. This was such a wonderful talk and definitely super needed. You know, we're entering mushroom season. You see this stuff everywhere. If you guys got questions, you can pipe up or post them in the chat. I'm happy to help read them out here. No pressure as well. I think we'll start with, let's see, Sydney's question here. Do we have any examples of heavily invasive fungi that is further along the path that GOM seems to be going? And if so, is there any info on what the future might look like in regards to GOM? Just wondering if there are areas that have seemed to show great biodiversity loss where this has happened before in regards to fungus.
Yeah, Sydney, that's an awesome question. And um, basically, there are invasive fungi that are further along. I would say One really good example is the orange ping pong bat fungus. It's Favelacia cloudopus. It's like a bright orange fungus, and it's got a bunch of little holes in it. And it might have been from around Madagascar, but the unfortunately like the DNA and the population genomics is not conclusive, so we don't even really know where it started from. But it went crazy in Australia and especially in New Zealand. And it can grow on like 50 different types of wood, which is crazy to think about. I mean, golden oysters, I'm starting to keep track of how many different types of wood it can grow on, but we're not at 50. And so this Favelacia cladopus is now on every single continent. In fact, it is just beginning its invasion in the Bay Area. And I emailed some folks over there. I don't know what's gonna happen, but so that's one thing. And then I think there are also non-wood decay invasive fungi like Amanita muscaria, definitely on every continent except Antarctica now. So further along in that geographic spread also. But the other part of it, what's happening to biodiversity loss, the crazy thing is, either because it's been difficult to study, my publication will be the first one to show biodiversity loss being caused in nature by an invasive fungus. And so I'm obviously really excited about that, but I have looked over and over to be like, this can't be the first time someone's showing that. But basically, like the studies with co-invasion, they've done a good job at like trying to see biodiversity loss, but I think it's been more from the plant perspective. And so yeah, so co-invasions where a tree invades along with the mushroom are harder ones to kind of disentangle because we don't know if the fungal loss is due to the tree or due to the fungus, right? But this is the first case where it's just a fungus causing the biodiversity loss of other fungi. So yeah, that's my answer.
All right. Well, we got some more questions here. Does anyone want to pipe up? Otherwise, I'll take one from the chat again. All right. So Sarah asks, speaking of cultivation and mushroom grow kits, my mind went to oyster types that are commonly available, like blue and or pink varieties. Why are we not seeing these in the wild yet? If we are not seeing those in the wild, what's so special about the goldens?
I love the pun. Thank you for
Mm,
that.
you did not do this justice. Not sure this can be easily answered, but food for thought.
Yeah, I've thought about this too, and like my only answer, okay, my first answer is I don't know. My second answer is I really think it's just because of the similarity in overall climate type and ecosystem type to the native range of like Russia, China, Japan. Um, and yeah, and so as for the blue or pink varieties, I have I unfortunately have heard that the pink variety might be becoming invasive in New Zealand. I have a very sweet pen pal from New Zealand, David Hara, who just completed his PhD and defended. And he is also working on golden oy or sorry, he's working on oyster mushrooms and basically disentangling a couple different species to try and make it clear which ones are native and which ones are not. Um, so I think that the other ones are capable of being invasive, especially if they're cultivated, because I think cultivation has a lot to do with making species cold hardy and resistant to molds and other microbes, heat tolerant. Like, right, there's a bunch of things where basically cultivation can help groom it into being a really strong invasive. So we might not see the blue or pink here, but I think the yellow one, because of its native conditions, is doing well here.
I want to jump in here and say, when I was in China, Aishwarya knows this, but I went to talk to people who know about golden oysters that are native to China, and they say like golden oyster is a pretty rare mushroom to find in the woods there, you know. So it's possible that since it's been domesticated, since it's been brought over, something has changed in its biology, or maybe there's like some biocontrol, something or other that's happening that's keeping its population numbers down. All right, we got.
Yeah. Oh, and sorry, Jason, I'll just add to that. To be clear, that is what my next chapter, the population genomics one, where I'm asking folks to send me golden oyster samples from all over North America. I'm also getting ones from Asia, and so I'm going to try and compare their genomes, their genetics, and see if there are places where their DNA has evolved, been artificially selected for, or changed in some way compared to the native range. It's gonna be bananas cool.
Can't wait. All right, we got 10 minutes left here, and we got a ton of questions, so we're gonna try and speed round the rest of these. So, what species of trees did you sample, and did GOM seem to displace fungi in some trees more than others?
I sampled um 22 of the trees were American elm, Ulmus americana, and then four of the trees were Ulmus rubra, the slippery elm or the red elm. So no, I didn't notice a difference based on species, but to be fair, that's not like a fair sampling of comparing how it's doing in red elms versus American elms.
Cool. Rachel says that GOM is displacing elm and oyster in southeastern Michigan, and or no, elm, oyster, and enoki in southeastern Michigan. Kind of echoing what she said.
Yikes. Send,
Um,
send me a sample, Rachel. Send me.
yeah. If you missed the link, I put it in the chat earlier. It's also on the Pringle Lab website. All of you guys, you can contribute to Aishwarya's super important project. So, you know. And I think is there a reward, Aishwarya, for participation?
The reward is a surprise at the end, but because y'all are my Madison crew, I will share it to you. I want to create an original artwork, art piece and send that out to everyone. So yeah. So I am taking down everyone's contact information who've sent stuff to me.
There you go. Get in on the ground floor. All right. Jen says, forgive me if I missed this earlier, but how do you differentiate from fungi being displaced by GOM from fungi that might be competitive with GOM or make a tree more resistant to GOM infection? Have you found any species that are perhaps protective against GOM? Fantastic question.
Yeah, that's a really good question. I'm going to answer what I interpret you're asking. So, for every fungal species, I I made a proportion of how many times that species occurred in my data set of all of the wood shavings I took, right? So like let's just say Serena unicolor, I will have seen that it appeared, let's say 86% in 86% of the non-GOM trees, but it only occurred in, let's say, 40% of the GOM trees. So I created like a percentage or a proportion of how prevalent each fungus is. And then I did like a statistical test to see if that number, that difference between 86% and 40%, whether that was significantly different or like a significant drop. And so that is the way that I kind of tested which ones are being displaced, whether or versus which ones are not, right? So there was some amount of fungi that didn't show much of a change in percentage or proportion. Um, but then kind of like the other nature of your question, right? Like how can I tell them apart from fungi that might compete with GOM or make a tree more resistant? I can't tell. I can't tell the difference between that, right? I have to assume that every fungus in there is competing with GOM for its life, for its territory. Um, and that some of those fungi might also maybe they have a role in making a tree resistant um to GOM, but yeah, but it really is I just don't know.
Hmm.
Yeah. Also, let me just grab my back battery real quick, but I can still hear you.
All right, you got it. So I'll answer a question for Dennis here asking, can I get the link to the recording of this presentation? Yes, anyone who wants the recording of this presentation, I may, your humble host, have forgotten to press record until like 5 minutes in, but everything other than the first 5 minutes will be posted on the MMS website, and I believe we have a newsletter, so just keep an eye out. You should find, or you should hear something about it coming up. We have a whole speaker series, so hopefully we're gonna have all of this, uh, you know all in the same place, so it'll be easy for people to reach and view and share and so on and so forth. All right.
I love Rachel's next question. Maybe it has an antimicrobial that our bacterial populations are unfit to compete with. Maybe there is a new antibiotic potential. Absolutely. So you just hit the nail on the head with this theory in ecology called um, like the novel weapons hypothesis. And it's one of the ideas that with invasive fungi, if they have some sort of new novel weapon in the range that has never experienced it yet, they don't know how to fight against that weapon. So yeah, it's possible.
How does gum affect other native species of Pleurotus? Is that not well understood yet? And why are native oysters not as aggressive as gum?
Gosh, I wish I could tell you, but I have no idea. I think I've had the same experience in Wisconsin, and a little bit in Minnesota. Like I've seen native oysters around, but they are not growing at all the same way that golden oysters do. And the kind of interesting thing is I worked with a recently graduated undergrad, so congratulations to Zoe. She did a little project in our lab where she grew different species of Pleurotus, different oyster mushrooms, like the native one, Pulmonarius or Ostreatus. She plated them on agar together with gum, and actually the native oysters won. So that was kind of really interesting to see. We don't know if that still applies in the wood environment, though, right? Like the kind of frustrating thing about science is you can show something happens in a lab, but is that necessarily the same as it happening out there in nature? We don't know. The wood environment is completely different than an agar plate kept nice and warm in an incubator. But like generally, I can tell you that on these agar, sugar media plates, the native oysters grow way faster than golden oyster does. So kind of interesting.
Yeah, super
I don't know. Someone will find out someday.
fun. Hopefully you. All right. Couple more questions here. Great question from Garrett. Are there alternatives that are native to Wisconsin? How can we you know enjoy oyster mushrooms that aren't goldens and pinks and blues?
Yeah, I think the best I think the best alternative is if you find a native oyster out there in the woods, take that, come home, culture it, or like put it into a wood log or whatever. Like that is the most foolproof way to like if you want to have a grow kit or like, kind of culture mushrooms in your home, um, getting one from that you have foraged, that you have a general sense is native to your area is kind of like the best recommendation. As far as kind of like grow. Yeah, I guess really what I'm saying is that I can't say that I trust grow kits anymore, you know? Like you just you just don't know what those strains have gone through. Maybe they've been artificially selected. Um, yeah, yeah, that's my thought on that. And like I hope it's clear though that it's totally fine to I encourage everyone to just forage and eat all the golden oyster you can, right? It's here. That cat is out of the bag. So you might as well enjoy it.
Mhm. Destroy it with your stomach acid. It is your civic duty. Um, well, we got like 1 minute left. One last comment. Oh my gosh, it's super long.
Oh, wow. I hope you're doing okay in LA.
Okay, well, you guys are welcome to hang on here for audience members. I will be hanging out for a little while longer. Make sure you check out the future talks for MMS. We got more stuff on the calendar here, lots of interesting folks. Also, another Pringle Lab member coming around eventually, the fantastic Cecilia Stokes. Not to steal your thunder, Aishwarya.
No, no, that's totally fine.
So yeah, make sure you tune in and you tell your friends. And yeah, thank you so much, Aishwarya. You have any closing thoughts or something you want to say before we wrap this up?
Yeah, just I guess a quick comment to Sarah, who's done the reverse journey of me, where she used to live in Madison and is now in Southern California. I'm so glad that you're finding the diversity of mushrooms there, you know, like so when I moved back from New York to Southern California, it's really that once I knew to look for them and like maybe where to go look for them, I started to find fungi everywhere. So I think there's really something to be said of like once you know a thing and once you know its name, you start to see it, you know, and you start to find it around. And also give my love to the lambs, folks. I used to be part of the LA Mycological Society too. That was the first like mycological group I joined and they were yeah, just really great. So I'm glad that you're having fun with that. And I suppose my last parting thought is like, this is nobody's fault. If you know, like a mushroom grower, it's really on my mind because I just got a long email from a mushroom grower today who was like, I got my bachelor's in ecology and I'm a mushroom grower and golden oyster is like one of my best sellers. And like, I've been hearing more people talk about how invasive it is. Like, what do I do? And, it's a great question. And it kind of weaves into how excited I am to approach the science communication part of this work, right? Like us coming together and talking about how to deal with this situation is the most important thing. It doesn't matter who brought it here. We didn't know. We didn't know that invasive fungi could do this. Like I know other mycologists I've worked with who didn't know that fungi could be invasive. So from every level, from academics, to yeah, basically to anybody, this is all something that we're learning together, and there's no fault to be had, only learning. So,
Wow, wonderful. Well, when your paper does come out, how can people find it? How can people find you? And so on and so forth.
you can check out the Pringle Lab website for sure, but I will definitely also send the link to the publication through Jason to y'all's newsletter.
All right. Well, thank you once again, Aishwarya, and thank you everyone for coming. That is a wrap. Whoop, whoop. Great job.
Thanks. Feel free to send me an email. I'll hang around if anyone has last things they want to talk about.
Oh,
If you have a second, could you explain the, was it NMDS measure that you use?
Yeah, sure. I used the oh man, I just looked this up earlier today. I used the Rop Crick distance matrix, distance metric in my NMDS. Um, and I used ROP Crick because it's been shown to be like appropriate for zero weighted data sets. So when I get back the Illumina reads, I convert all of those read numbers into like binary, like present one and absent zero. And so, from some other mycologists that have worked on community ecology like this, they recommended that ROP Crick would be a good distance metric to use. Do you have thoughts on that?
Oh, it's pretty far over my head. I was just kind of curious of like the basic construction.
Oh, um, uh,
So like if I can ask it another way, so it is based on just a matrix, like a fully binary matrix of the presence of DNA of different species.
yeah. Yep.
Okay,
Yeah.
interesting.
Yeah, yeah. So each data point on my NMDS, which like when you see the figure in the publication, it's not like the one that I showed where it's like just an oval. You'll see every dot, and every dot is a fungal community from a given height in a tree. And then, yeah, and what is encoded into that community is the presence and absence of the presence or absence of each OTU, of each fungus.
So then to get from the point where you have that to the coordinate on the graph for each, so you know the height one on tree A, how do you get that coordinate?
Well, that's computer magic.
Okay.
Yeah, no, I basically just like I run this line of code, right? In our studio. And so like, I don't know that I can explain it much more than maybe like, than your understanding of it. I know that the computer calculates a distance metric for each of these points and how dissimilar they are, but like I don't know the exact equations of how to do that or how to yield the coordinate myself.
Okay. So it's not like if I say, oh, it's the coordinate is, you know, 1, 2, that is meaningful independently.
Correct. Exactly. It's
Okay.
yeah, yeah.
Okay.
Yeah, yeah. It's not meaningful independently. You're exactly right. Because the axes, this is actually something that really bugs my husband because like he's also a scientist, but the axes are just NMDS axis 1 and NMDS axis 2 and he's like that doesn't mean anything. And indeed, it doesn't mean anything. It's just coordinate space for the computer to use.
Okay. Okay. I think that makes a little more sense. Thank you.
Yeah.
Anyone else have any comments? I want to pitch in? I got a question for you if no one else wants to hop in here. So when are we gonna get a sporeless variety of gum?
You tell me, Jason, you tell me. Yeah, I was just thinking about that today because the mushroom seller that emailed me, he mentioned that and he was like, what's the best way to resolve this situation? And yeah, there's a guy that I met in Europe at the conference. I think I showed you that email, and his lab specializes in making sporeless varieties of mushrooms, and it definitely already exists. Like the roadmap to making a sporeless variety absolutely already exists. Someone just has to do it. So I was actually gonna reach out to that guy in Europe and ask him, "Hey, do you want to do this?"
Ooh, can't
They
wait.
are tasty.
Yeah. So, what do you like to do when you're not giving Zoom talks here?
What do I like to do? I've been playing Age of Empires, nostalgic, a really nostalgic game. Um, where you just kind of like go through the battles in history and kind of just redo them, you build up a little civilization and stuff. Bye, Ben. Thank you. Um, yeah, and I don't know, hang out, go on walks, listen to music, play.
You're not gonna plug? Come on.
Oh, I should. Oh, yeah, for whomever is left, I'm in a band, and the name of the band is Big Slide. You can follow us on Instagram at Big Slide 4. And yeah, it's a super fun time. I really love it.
Yeah. When's your Golden Oyster song gonna come out? That's what I want to know.
Gotta get cracking on that.
Golden Oyster Blues. I'll listen to that. I'll drive up your Spotify listens every
Yeah, thanks.
It's all just Jason. This, dumb number one listen to.
Yeah.
one listener, a million listens.
Comes up in your Spotify Wrapped. You're the number one fan.
Heck yeah. All right. Well, if no one else has anything else, I know you're a busy grad student, so I don't want to say
Oh,
between you and
yeah, I guess sorry. I just saw a last question in the chat. Aiden said, "What's your favorite way to cook, eat gum?" I put them in the pan and I try and draw the water out of them, right? Kind of like with a lot of other mushrooms, that's the first step. Um, so I'll add a tiny bit of water to like get an initial amount of steam going. I'll cover it up with a lid. I'll try and draw all the water out of it. And then by that time, it's kind of starting to stick to the pan, and that's when like I'll add some olive oil or butter, and then like really saute them until they're nice and crispy, and then add garlic. You know the rest.
That sounds delicious. I'm eating some golden oysters for dinner tonight, so I might
Oh,
try
nice.
something.
Sweet, sweet. Yeah. The only bad thing is just the way it makes your kitchen smell. Like, yeah.
Yeah, that's just oysters though.
smells
Yeah.
better than actual oysters.
Yeah. Okay. All right. Well, thank you. Thank you all. Yeah, it was super fun. I hope I get to go on a foray with you all sometime soon.
Yeah. And
Yeah,
once you publish, maybe once you got super fancy results, you can come back and show us the slides that are top secret, you know?
Yeah, right. I know. I feel so dumb having to hold that in, but
Yeah.
orders are
All
rules
right.
are.
Okay, well,
Bye.
that's the end.